Hello everyone and welcome to my new session which is on modes of pollination and double fertilization. Myself, Dr. Narkade Gopal Vasudev, working as Assistant Professor, Department of Genetics and Plant Breeding, School of Agriculture, SR University, Warangal. So in today's session, we will see about what is pollination, what are the different mechanisms of pollination promoting self and cross pollination, and what is the concept of double fertilization. So one by one we will see. First we will see what is meant by pollination. So this pollination is transfer of pollen grains from the anther to the stigma of a flower. So like this in figure you can see in the picture, like transfer of pollen grains from anther to the stigma, right? These pollen grains are immobile. That means they cannot reach the stigma by themselves. That's why they are immobile. So an external agent is required for this. An external agent is required to take the pollen grain to the stigma. So these external agents may be wind, water, maybe animal, gravity or growth contact. So pollen grains are immobile. So they cannot reach the stigma by themselves. So that's why external agents like wind, water, animal, gravity or growth contact is needed to take the pollen from anther to stigma. So these are some terms related to sexuality of individual flowers. So first that is bisexual or perfect flowers. The flower with both male, that is androecium, and female, that is gynoecium reproductive structure. That is including stamens and ovaries. That is called as bisexual flower. Flowers that contain both androecium and gynoecium are called as hermaphroditic. That is, or else you can say like hermaphrodite flower. So, bisexual flower is also called as hermaphrodite flower. So, example of plants with perfect or bisexual flowers will include like lily, rose. A complete flower is a perfect flower with petals and sepals. So now like you may have aware like four floral rolls are there: calyx, corolla, androecium, gynoecium. So when in a flower androecium and gynoecium both are present, that is called as bisexual flower, right? If either male or female part is present, that is called as unisexual flower, okay, or imperfect flower or incomplete flower. So single member of calyx, that is called as sepal, single member of corolla, that is called as petals. And in some condition, when calyx and corolla both are fused, so that floral roll is called as perianth, right? So these are the terminologies with related to sexuality of individual flowers. Okay. So see, these are the this is the flow chart which will is showing the classification of pollination. So mainly two types of pollinations are there. One is self-pollination, second is cross-pollination, or you can say xenogamy. So in self-pollination, autogamy and geitonogamy. So autogamy means transfer of pollen from anther to stigma of same flower, right? And what is geitonogamy? Geitonogamy means transfer of anther from one flower to the stigma of another flower but of same plant. That is called as geitonogamy. So just concentrate on the difference. Like autogamy means transfer of pollen from anther to stigma of same flower, right? And geitonogamy means transfer of anthers from one flower to the stigma of another flower but of same plant. That is also, it is one of the type of self-pollination. And second is cross-pollination. You can also call as xenogamy. So there are two major agencies like abiotic agencies and biotic agencies. So in abiotic agencies, anemophily, that is pollination through wind, and second is hydrophily, that is pollination through water. Right? Just now we have seen like pollen grains are immobile. So external agents are required to carry them from anthers to stigma. So likewise, in abiotic agencies, anemophily, that is pollination by wind, and hydrophily, that is pollination by water. And second type, that is biotic agencies. So there are four different insects are there. Like first, through insects, that is entomophily; through bats, that is chiropterophily; through snails, that is malacophily; and last, birds, that is ornithophily. So this is the brief classification of pollination. So now first we will see what is meant by self-pollination. So this self-pollination is the transfer of pollen grains from anther of a flower to the stigma of either same or genetically similar flower. So self-pollination, once again I will repeat, that is transfer of pollen grains from anther of a flower to the stigma of either the same or genetically similar flower. So accordingly, the self-pollination, it is of two types, that is autogamy and geitonogamy. So here you can see in the picture, like see from anther to stigma. So these are the pollen grains, right? So here also you can see these pollen grains from anther to stigma. This is called as self-pollination. So multiplication of plant by fertilized embryo, that's self. So first autogamy. So this development of seed by self-pollination. So now we will see some mechanism which will promote the self-pollination, or you can say which will promote the autogamy. In that, first is bisexuality. So we have already seen that bisexuality means presence of male and female organs in a same flower. So in this, for example, sunflower, hibiscus, lily and mustard. Then second mechanism is homogamy. So in this case, in the case homogamy means maturation of anthers and stigma of a flower at same time. And suppose in one flower anthers and stigma are there, so both will mature at same time. That is called as homogamy. So maturation of these two at same time means ultimately it will promote self-pollination. So in this case, examples are wheat, rice, mirabilis, potato and sunflower. Then third mechanism, that is cleistogamy. So then what is cleistogamy? It is a condition where pollination and fertilization will occur in an unopened flower bud. So before opening of flower bud, pollination and fertilization will take place. That is called as cleistogamy. Example, wheat, barley, oats. So then fourth, that is chasmogamy. So what is chasmogamy? So it is a condition in plant where fertilization occurs after opening of flower. That is called as chasmogamy. Examples, wheat, barley, rice, oats. Then last mechanism, that is position of anthers. So what is this position of anthers means what? That is stigmas are surrounded by the anthers. So when stigmas are surrounded by the anthers, ultimately it will ensure the self-pollination. So this case you will find in tomato and brinjal, right? And again second condition is when stamens and stigmas are enclosed by the petals. So means when corolla will cover the reproductive part. So this kind of things you will find in green gram, black gram, even in soybean and chickpea. So these are some mechanisms which will promote the autogamy. Now next, that is geitonogamy. So this is also one of the type of self-pollination. So this geitonogamy is a type of pollination in which pollen grains of one flower are transferred to the stigma of another flower belonging to either same plant or genetically same plant. So transfer of pollen grain from one flower to the stigma of another flower belonging to either same plant or genetically similar plant. That is called as geitonogamy. So in geitonogamy, the flowers often show modification similar to ones found in cross-pollination. Because see, the definition itself indicates like transfer of pollen from one flower of same to the stigma of another flower or of same plant or genetically similar plant. So that means pollination between two different plants. Though both the plants are genetically similar, so it is somewhat like cross pollination. Okay. Now what are the advantages? So it maintains the parental characters or purity of the race. So this self-pollination is used to maintain the pure lines for hybridization experiments. The plant does not need to produce large number of pollen grains. Right? The flowers do not develop devices for attracting the insect pollinators. What are the disadvantages? The new useful characters are seldom introduced. So during the pollination and fertilization, automatically some new useful characters will be introduced. Vigor and vitality of the race decreases with prolonged self-pollination. Immunity to diseases decreases. Adaptability to change environment are reduced. Then the next we will see cross-pollination. That is also, it is also called as xenogamy or allogamy. So cross-pollination is the transfer of pollen grains from anther of one flower to the stigma of genetically different flower. Cross-pollination is the transfer of pollen grains from the anther of one flower to the stigma of genetically different flower. Right. So cross-pollination is performed with the help of external agencies like we have seen during classification. So again, the agencies like we have seen pollination through wind, that is anemophily. Pollination through water, that is hydrophily. Some biotic agents like through insect, entomophily, through birds, ornithophily, through bats, chiropterophily, through snails, malacophily, through ants, myrmecophily, and through snakes, ophiophily. So these are some terms related given to the agencies of the pollination. So first we will see what is anemophily. So this anemophily, it is the mode of cross-pollination or transfer of pollen grains through the agency wind. So for example, here the plants like coconut palm, date palm, maize, many grasses and cannabis. What are the characteristics here? So the flowers are colorless, odorless and nectarless. The pollen grains are light, small and winged or dusty, dry, smooth, non-sticky and unwettable. So these are the characteristics for that pollen grain. The stigma is hairy, feathery or branched to catch the wind-borne pollen grain. The pollen grains are produced in a very large number, right? Next, that is hydrophily. That means mode of pollination is water or transfer of pollen grains through water. So, example, Zostera and Vallisneria. Then in this case, what are the characteristics? Flowers are small and inconspicuous. Nectar and odor are absent. Pollen grains are light and unwettable due to the presence of mucilage cover. Stigma is long, sticky but unwettable. So these are some characteristics in case of hydrophily. Then third mechanism, that is entomophily. That is the pollen grains are transferred to a mature stigma through the agency of insects like moths, butterfly, wasps, bees, beetles, right? The characteristics are flowers are showy or brightly colored. Most insect pollinated flowers have landing platform. Pollen grains are spiny, heavy and surrounded by yellow oily sticky substance called pollen kit. Stigmas are often inserted and sticky. Some flowers provide a safe place for insects for laying the eggs. So next mechanism, ornithophily. So it is the mode of allogamy performed by birds. Only few types of birds are specialized for this. So they usually have small size and long beaks. What are the characters? Ornithophilous flowers are usually bright colored. That is red, orange, yellow or sometimes blue. The floral parts are commonly leathery. Ornithophilous flowers secrete abundant watery nectar or have edible parts. Scent is often absent. Then we have seen agencies for cross-pollination. Now we will see the mechanism to ensure the cross-pollination. Right. So there are different types of mechanism. That is, first one is dicliny. That means plants are with unisexual flower. So what is unisexual flower? Either male parent or female parent. Again this dicliny, it is of two types. First, monoicy. In monoicy, male and female flowers are separate but on the same plant. That is called as monoicy. These male and female flowers are present in the same inflorescence or sometimes on separate inflorescence but on the same plant. That is called as monoicy. So for example, when male and female flowers are present in same inflorescence, in that case examples are mango, castor, banana. When male and female flowers are on separate inflorescence, then examples are maize, cucurbits, grapes, strawberry and rubber. Then second type of dioicy, that is staminate and pistillate flowers are present but on different plants. That is, male plant is separate and female plant is separate. So in those cases examples are like papaya, date palm, spinach, hemp and asparagus. Then second type, likewise we have seen homogamy in case of self-pollination. So vice versa, dichogamy is there in cross-pollination. So what is this dichogamy means? So homogamy, maturation of anthers and stigma at same time. Okay. So conversely, dichogamy means maturation of anthers and stigma of a same flower but at different times. That is called as dichogamy. So this dichogamy, as the anther and stigma mature at different times, On the basis of that, again dichogamy is of two types. So first one, protandry. That means maturation of anthers before stigma. So in this case, examples maize and sugar beet. So second, protogamy. That means maturation of stigma before anther. So example, protogamy. Example of protogamy, that is pearl millet. Then third mechanism is heterostyly. As the name itself indicates, the length of style and filaments are different. Right, so it will control the self-pollination. So and it will automatically promote the cross-pollination. Example is linseed. Fourth mechanism, that is herkogamy. So what is herkogamy? That is in hindrance to self-pollination due to physical barriers such as presence of hyaline membrane around the anther. So for example, alfalfa. Fifth mechanism, that is self-incompatibility. That is inability of fertile pollen to fertilize the same flower. So example for self-incompatibility, that is brassica, radish and nicotiana. So these are some examples. Last mechanism, that is most important, male sterility. That is presence of non-functional pollen grains. So for example, that is sorghum, wheat, okra in case of vegetable and rice. What are the advantages of cross-pollination? So this cross-pollination introduces the genetic recombinations and hence variations in the progeny. So cross-pollination increases the adaptability of the offspring towards the changes in the environment. So it makes the organisms better fitted in the struggle for existence. Right. Then the plants produced through cross-pollination are more resistant to diseases. Then what are the disadvantages? A factor of chance is always involved in cross-pollination. This cross-pollination is less economical. Some undesirable characters may come into the race. The very good characters of the particular race may likely to be spoiled. Now, what is the significance of pollination? We have seen self-pollination, cross-pollination. Then what is the significance? So the mode of pollination plays a very important role in plant breeding. So it has impact on 5 important aspects. So first foremost aspect, that is gene action. Second, genetic constitution. Then third, adaptability. Fourth, genetic purity. And last, that is transfer of genes. The percentage of cross-pollination can be determined by growing a seed mixture. The percent of cross-pollination you can determine when by growing seed mixture of 2 different varieties together. The 2 varieties should have marker characters, say green and pigmented plants. The seeds are harvested from recessive variety and grown in the next year in separate field. The proportion of pigmented plants in green variety will indicate the percentage of outcrossing or cross-pollination. So that's how you can calculate the percentage of cross-pollination. So that is all about the modes of pollination. So our next topic, like as per the title, that is modes of pollination and double fertilization. So now we will move towards double fertilization. Okay. So now what is fertilization means what? So plant fertilization is the union of male and female gametes to produce fertilized egg, that is zygote. Then what is double fertilization means what? Both the male gametes participate in sexual reproduction. 2 male gametes fuse with 1 female gamete wherein One male gamete fertilizes the egg to form zygote, whereas the other fuses with the two polar nuclei to form an endosperm. Triple fusion is the fusion of male gamete with two polar nuclei inside the embryo sac of the angiosperm. So this double fertilization is a unique and universal phenomenon in the life cycle of angiosperm. It involves two separate nuclear fusion events and therefore it is called as double fertilization. First is the fusion between female gamete or egg and one of the male gametes to form a diploid zygote. That process is called as syngamy. The second fusion is between the other male gamete and the secondary nucleus, leading to the formation of triploid primary endosperm nucleus. That's why the whole process is called as triple fusion. Right. The zygote develops into embryo and the pen, that is primary endosperm nucleus, in short form we have given pen, forms the endosperm. So this phenomenon was first reported by Navaschin in 1898 and Guignard 1899 independently in Lilium martagon and Fritillaria tenella of the family Liliaceae. However, the term double fertilization was given by Thomas in the year 1900. The rudimentary fertilization was also reported in Ephedra and Natum. So what is the background? Now we will see the background of double fertilization. The transfer of pollen into the stigma of pistil is a prerequisite for the fertilization. So actually this is known as pollination, which is of two types, self and cross, we have seen. So the double fertilization is a complex process requiring coordinated action of the component cells. Recently, the plant embryologist extensively studied these series events. With regard to this, there are two concepts, that is male germ unit and the female germ unit, proposed by Dumas et al. In 1984. So now, this is the stepwise events in double fertilization. So now see, these are the parts, like this is the pollen grain, then this is the stigma, style, this complete is ovary, these are the ovules. So inside ovule, eggs and polar nuclei. So here you can see the enlarged version, this is the egg cell, these two are the synergids, these are the sperm nuclei, these two are the polar nuclei, and the bottom three antipodal cells are there, and this end is micropyle, and this is the pollen tube. So like this, here you can see the pollen growth of pollen tube, right? See the first, the pollen grains adheres to the stigma which contain two cells, one is generative cell and one is tube cell. The pollen tube cell grows into the style, just you can see the pollen tube grows into the style, the generative cells travels inside the pollen tube, it divides to form two sperm nuclei. The pollen tube penetrates an opening in the ovule through micropyle. So one of the sperm fertilizes the egg to form diploid zygote and the other sperm fertilizes with two polar nuclei to form the triploid endosperm, which will become a food source for the growing embryo. Then now types of fertilization based on the path of pollen tube entry into the ovule. Right. So first, porogamy. So what is porogamy? Entry of pollen tube through micropyle. Then next, chalazogamy. Entry of pollen tube through chalaza. Then mesogamy. Entry through middle part are the integuments. So integuments mean this outer line, outer cover. This is called as integument. Now steps leading to the fertilization. So first, germination of the pollen grain. So Stigma function is to provide the place of lodging and germination of the pollen grain after the pollination. So there are two types of stigma, that is wet stigma and dry stigma. So what is wet stigma? It secretes the exudates like water and other nutrients in the form of droplets on the stigma. Exudates made up of mix of water, lipids, sugars, amino acids, phenolic compounds. High viscous and adhesive, for example, petunia. And dry stigma means do not secrete any exudates. For example, Gossypium species. Germination of pollen may occur immediately or may take few days to weeks. Exine imbibes water and swells up, and in time develops into pollen tube and come outside of the germ pore. This exine and intines are the protective layer of the pollen. Then monosiphonous, example Campanulaceae, Cucurbitaceae family, Malvaceae family. Then polysiphonous, that is Althea rosea, Malva neglecta, then branch Acelpiadaceae and Archidaceae. Then masses of pollinia, that is grouped pollen. Many pollen tubes germinate simultaneously. Pollen tube carries two male gametes. Second step, that is movement of pollen tube inside the style. So pollen tube reaches the style through stigmatic papillae, solid or close. The tip of the pollen tube secretes hydrolyzing enzymes, that is pectinase. Example, dicots. So in monocots it is hollow and open. So third case, that is semi-solid or half closed. The content of the pollen grain reaches to the pollen tube tip. It is unidirectional, enters through micropyle. Entry into the embryo sac through piliform apparatus. Then third, that is pollen tube in the synergids. Entry only through micropyle, guided by arbiter. The presence of chemotropic substances. Collapse of one of the synergids prior to the entry of pollen tube. Discharge of pollen tube content. Two male gametes, vegetative nucleus and cytoplasm into the synergids. Then disorganization of the tube nucleus. Fourth step is double fertilization and triple fusion. Both the male gametes are involved in the fertilization. So fertilize two different components of the embryo sac. That's why name is double fertilization. One male gamete fused with egg nucleus, that is called as syngamy, to produce a diploid zygote. Another male gamete fuses with the polar nuclei to form triploid primary endosperm nucleus. It involves fusion of three nuclei, that is why it is called as triple fusion. Next is polyspermy and heterofertilization. So what is this heterofertilization means what? Type of double fertilization in plant in which endosperm and embryo are genetically different. So this happens when two different sperm nuclei from two different pollen tubes happens to enter into the same embryo sac. What is the significance of double fertilization? So it gives stimulus to the plant, due to which ovary develops into fruit and ovules develops into seed. It restores the diploid condition by fusion of haploid male and female gametes. It results in the formation of diploid zygote, which develops into an embryo and gives rise to new plant. So it results in the formation of triploid primary endosperm nucleus, which develops into endosperm in the seed. It provides nourishment to the plant. That means it provides nourishment to the developing embryo. It brings about recombination of characters, resulting in variation among the offsprings. So this is all about the double fertilization. So in this session we have seen modes of pollination and double fertilization. So hope you will get the information through this session. Okay, so till next session, thank you very much. Thank you.